Hello, everybody. So, that's a title that killed. No-kill science. Why and what for? This is actually the protocol we used and we still use in Eurosync studies to assess morphology and genetic of the seahorse and pipefish species in Europe. I'm going to talk about, I need to find out how it works, yeah. About the protocol itself and the way we work, and give you a few examples afterwards, if I still have time. So, of course, you've watched the photos there, and you noticed that there are a few individuals that don't fit in the general scheme. These are hippocampus. This is Gutulatus from the Tow Lagoon, and these here and here are Gutulatus from the Mediterranean Sea, so marine versus lagoonal. Okay, so why study morphology? Because morphology varies a lot, and this is one of the things we have that actually triggered the Eurosync project. How does it work? How does it change and vary? And how can this evolve? And what is it driven by? So here you can see that this. Look, yeah, okay, I know that I shouldn't have done that. Okay, so this is where I should push the button. So you can see that all this is supposed to be the same species, but with an impressive variability in aspect. This is also the same for the Acus type pipefish. So this is what we have to sort out without killing too many of them. So the usual way of studying morphology is either counting some like number of fin rays, things like that. This is what we call meristic characters, and you have metric characters which are measuring something. And usually this is expressed in terms of percent. For instance, the depth of snout compared to the length of snout, or compared to hand length, or things like that. So the use, as I said, of course, morphology has been worked on for a long time. And we usually use preserved animals. Preserved, that means dead. So we used to work on dead bodies. And the idea is, why not work with live animal and let them live afterwards? This is why we preferred the idea of to make photos with very interesting subjects for the photo which Sylvie took. And how we do proceed, so this has been discussed at the onset of the Eurosync project with all the partners. The idea is to have a series of very specific photos, which I won't list, but it deals with head shape, with body features like number of body rings, at least trunk rings and dorsal fin rays. There are possibly issues with the shape of the tail, and also, as Nuno showed you earlier, if we can have a good photo of the whole fish, this can help for some interesting questions. So with that, we have a series of photos, and what is important to me is that we must deal with that as with we did before with specimens. There are reference specimens that we can use for future study that could be stored in museums and so on. So it has to be done very seriously. This is the testimony of one individual to science. So what we do with that is we can, there are many things we can do. What will be done a lot with Eurosync is not the classical approach, which I told you about, the proportion of this dimension and that one, but a more, I was about to say automatic, but it's not automatic. There's a lot of work in between, but there's a way to be able to deal with the shapes in a mathematical way. This is, how is it called? Geometric morphometry. And this is what has been done, for instance, for Syngnathids tiflae, or the ones which look like that. The numbers in red are landmarks. So we take a photo with the specific software. We put the landmarks in the right place on the photo. And we have coordinates for all these landmarks, which help us show a shape. And you also have the blue lines, which are more, they are not precise points, but a series of points along the side, the upper side and lower side of the head. So with all that, we are able to build a figure, which is the image, the electronic image of the fish. Here we had 15 landmarks, but some species lack some of the reference points, like no pectoral fin in Neurophis, like you can see here. So Neurophis has 2 landmarks less. And seahorses have more landmarks because we need especially to deal with the spines or protruding parts of the profile to have a more precise comparison between individuals, populations, and species. So this gives us some matter to work on morphology, either by working on what kind of change in shape you have along this gradient here, for instance, or it can be per population. We can have a mean image for each population, which allows us to say that they all have thin snouts, while those have thick snouts, or thicker than the others. So this is an idea of what we can do and how we manage. Of course, we don't forget more usual parameters, like dorsal fin rays, number of trunk rings, and caudal fin shape. As I told you, it can be interesting. for some species or populations. Okay. Yeah, still in the idea to be non-impacting on the fish, when we work on genetics, we need tissue samples, and of course we have an approach which is to minimize the size of tissue samples, even though geneticians might need just a bit more. But well, this is a way to go forward without impacting too much the animals. And well, I can swear they recover well when we do that. One example now. You have here on the left, for you, long-snouted seahorses from Thau Lagoon in southern France. On the right, you have the same species, but individuals from the sea, the open sea, so lagoonal versus marine. And we can use photos to, well, this is not very important, but this point here is the only place where you have the lagoonal Mediterranean lineage. So what we did, we worked with a series of the landmarks and tried to see if it worked well to represent the shape. And you see that it's kind of Zorro mask is quite a good representation. We also noticed that there can be some changes or some differences, which actually, in that case, are only due to the position of the head. Either it's bent or erected, and it changes some things, while it doesn't change some more important features, like, well, you see the area of the spines here. It's exactly the same shape, just there's a change in curvature here, which makes it more difficult to see. And by the way, the best thing we found was more traditional, which since we found some physical differences between the two populations. Sorry. With the Thau Lagoon ones with two spines on the cheek, and the nose spine, which is squarish, while the marine one has only one spine, one cheek spine, which is like very erect, and the nose spine, which is a bit thinner at the end. So you can tell where they come from when you see the fish, most of the time. Another example is what we call the mystery pipefish from the northwest Mediterranean. We don't know who it is, and we wonder if it could be a population of Syngnathids rostellatus, which is known to be Atlantic, or if it has something to do with Syngnathids tenionotus, which is in the Adriatic Sea, or maybe it's something like a forgotten species which was identified as Syngnathids agassiz or agassizi. And for that, we still don't know. I tried to work a bit on two meristic characters, which are the number of dorsal fin rays or trunk rings. And well, it's not that clear, so I hope, since we still have a lot of individuals to study, I mean tens of individuals to study, and some genetic samples. We'll have to disentangle the thing with all these parameters together. And the last one is of not much interest here, since Miriam will talk about this later and much more in detail than what I could have done. So that's it. Thank you.